Good afternoon, everyone. Bon après-midi, mesdames et messieurs. So I'll start with the land acknowledgements first, but before I do that, I see quite a few of our researchers in here, so I'd like to introduce myself first. My name is Amir Asif. I serve as Vice President Research and Innovation at York University. I'm also a faculty member in Lassonde School of Engineering. I'm an electrical engineer by profession, and I can see some familiar faces and some not so familiar faces. So let me start with the land acknowledgement. Thank you for joining us for the 18th annual York Gairdner Lecture. Such a pleasure to welcome you in person after 2 years of online events. York University acknowledges its presence on the traditional territory of many indigenous nations. The area known as Toronto has been taken care by the Anishinaabe Nation, the Haudenosaunee Confederacy, and the Huron-Wendat. It is now home to many First Nation, Inuit, and Métis communities. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. York University is proud to be among us the 17 universities nationwide hosting a National Gairdner Lecture. Each Gairdner Lecture held across the country gives us, as scholars, researchers, students, and Canadians the opportunities to hear from the 2022 Canada Gairdner Award laureates, and this year another remarkable group of scientists are being honored. Those who are profoundly improving the health and healthcare outcomes of global populations through the discovery, application, and translation of biomedical and global health research. For more than 60 years, the Gairdner Foundation has recognized distinguished researchers and scholars who have demonstrated extraordinary scientific excellence and leadership in advancing fundamental research in human health that impacts patients and populations around the world. Actually, since 1957, when the first Gairdner Awards were granted, over 400 scientists have received a Canada Gairdner Award, and these are from 40 different countries. And what is amazing is that 96 have gone on to win the Nobel Prize. So every 1 out of 4 Gairdner laureate wins a Nobel Prize. In fact, a number of Nobel laureates have previously been York Gairdner Lectures as well. A few recent examples that come to my mind are Emanuela Charpentier and David Julius. And I think Ron Austin is here, and Ron has been associated with the Gairdner Award for a very long time, and I'm sure he remembers these speakers. And finally, Jennifer Doudna is a York Honorary Doctorate recipient as well. So today we are honored to welcome our two esteemed speakers, Dr. John Dick and Dr. Katalin Karikó. It is a pleasure to welcome these 2022 laureates to the York campus to speak to us about their research and to interact with faculty and trainees in an exciting day of science. We're delighted to host and have this opportunity to hear research seminars by Drs. Dick and Karikó, who will tell us about their research on stem cells and RNA biology and its importance in vaccine development and other potential therapeutic uses. Both of the areas of research to be discussed by laureate speakers are areas of research where York has an important presence. Dr. John Dick has received the 2022 Canada International Gairdner Award for the discovery and characterization of leukemic stem cells, providing insights into the understanding, diagnostics, and treatments of acute myeloid leukemia, or AML in short. Dr. Dick's findings offer clear direction for improving clinical outcomes in leukemia and potentially other cancers. Turning to Dr. Katalin Karikó, she is a researcher she's a researcher and a recognized biochemist. She has worked tirelessly on developing nucleoside-modified mRNA and lipid nanoparticle, LNP as it is called, the drug delivery, the foundational technologies for the highly effective COVID-19 mRNA vaccines that I'm sure all of us have it in our cells now. Dr. Karikó's work has enabled this vaccine and also has the potential to revolutionize the future delivery other therapeutics for viruses like influenza or HIV. So on behalf of the York University, I'm grateful for their attendance here today. We are so honored to have both Dr. Dick and Dr. Karikó join us for the York Gairdner Lecture. With that, I'm now going to welcome Gabriela Krivdova to do further introductions. Gabriela is a former exceptional York undergraduate biology and subsequently York MSc student. She went on to complete a PhD with Dr. Dick in April of 2022. I'm happy to welcome Gabriela back. She will introduce both our laureate speakers. Over to you, Gabriela.
Hello, everyone. I would like to echo the words of Amir in welcoming you to the 2022 Gairdner program at York University. My name is Gabriela Krivdova, and I'm a recent PhD graduate from John's lab. So I'm really honored to be here today, and it's a real privilege to be able to introduce these two outstanding scientists, Dr. John Dick and Dr. Katalin Karikó. Before introducing our speakers today, I would like to just take a moment to say a few words about myself. I completed my bachelor's and master's degree here at York University approximately 7 years ago. Returning back to the campus today has brought many pleasant memories. It was actually the undergraduate biology program here at York University that stimulated my interest in science and directed my career path towards becoming a scientist. During my undergraduate degree, I received 2 undergraduate and CERC research awards that enabled me to gain experience in wet lab research. Given my interest in molecular biology, immunology, and virology, I joined the lab of Dr. Kathy Hudak here in audience, where I completed my master's degree in 2015. So my training at York University really laid a foundation to my scientific career. So I therefore encourage all the young trainees in the audience to really reach out to the faculty for different positions, attend symposia and scientific meetings, and apply for awards and scholarships, as it might change your life as it did mine one day. So following completion of my master's degree, I then went on to pursue my PhD degree at the University of Toronto. My first rotation was in John's lab, and I was immediately fascinated by the innovative and rigorous experimental procedures carried out in the lab, including the in vivo assays for normal and malignant hematopoietic stem cells, single-cell in vitro clonogenic assays, and the variety of molecular and genetic analysis that are routinely done in John's lab. I am really grateful for John's mentorship, for him challenging me and encouraging me and supporting my ideas no matter how much they deviated from the established path. John genuinely enjoys talking to his students and discussing scientific ideas, and really he creates a lab atmosphere that feels much more like family than a workplace. So maybe what you don't know is that John had perhaps too much fun at one of our winter lab retreats, where he ended breaking his rib while he was tobogganing with his students. So I am especially delighted to introduce John here today. John was born and grew up on a farm in southern Manitoba, where he completed his PhD at the University of Manitoba. John moved to Toronto in 1984, where he completed his postdoctoral degree in Alan Bernstein's lab, which guided him towards the field of hematopoiesis and leukemia. John is a senior scientist and Canada Research Chair in Stem Cell Biology at the Princess Margaret Cancer Centre and professor at the Department of Molecular Genetics at the University of Toronto. He is a recipient of many prestigious awards, including the 2022 Gairdner International Award for the discovery, characterization of leukemic stem cells, providing insights into the understanding, diagnosis, and treatments of acute myeloid leukemia. John's discovery of leukemic stem cell established that not all cells within the cancer are equal, and provided direct evidence for the cancer stem cell model hypothesis. His work with the xenograph assay in purifying leukemia cells into leukemic stem cell and non-leukemic stem cell population transformed our views of the origin and nature of cancer and laid the foundation for new approaches to cancer therapy. John's discovery of leukemic stem cells not only transformed the leukemia and hematopoiesis field, but also changed our approach towards understanding of other human cancers such as colon, pancreas, and brain. So without further ado, please join me in welcoming Dr. John Dick to the stage. His talk is entitled, "What Makes a Stem Cell and How Does It Go Bad?"
Been talking so much, I'm just going to keep myself hydrated here. Well, it's a true pleasure to be here. It's been, I was trying to think, it was probably been, I did give a talk at York probably 20 years ago. So it's been a long time since I've been here, and the campus is remarkable and how it's thrived and grown. And you have a subway stop, which is fantastic, because I remember all the buses that used to go up the streets. So, pleasure to be here. A real pleasure to have Gabby introduce me. She's just been a terrific part of the ecosystem of the lab and making the environment what it is. And she's aside from being a tremendous scientist that she's developed into, she's just a great team player and a great citizen of the lab. And what some of you don't know is that she began a little developmental biology experiment about 2 months ago. So we have her here on mat leave. So she's combined all the different things that happen. And it's really important that females in science are supported because there's only one time in life that you can have this developmental biology experiment, and that's often the time when you're getting your training. And we have to have mechanisms that allow, our female scientists to thrive in all aspects, scientifically and in their families. So it's important that we can support as much as we can. Okay, so what I'm going to do, and I hadn't really known about the instructions for this, so I'm actually, Gairdner said, can you talk in Calgary, can you talk in Toronto, can you, talk at York, and they're all 30-minute talks, and, just give the same talk. So I'm going to give the same talk. I mean, I could have given another talk, but I asked Ron, and he said, no, give that one. So if those of you who heard the talk yesterday, either virtually or otherwise, you can either sit back and sleep for a little while, or I know I talk quite fast, and so maybe you'll actually pick up a few more things this time around by seeing it a second time. So what I wanted to do is to kind of give you a little bit of a trajectory. Actually, I should put my timer on here, because I have a habit of talking a little bit too long. So just hang on 1 second. Oops. Stopwatch, there we go. Okay. What I thought I would do is to give you a perspective of some of the work that got me standing on this stage last night and getting this medal. But I didn't just want to talk about old stuff, but I also wanted to give a flavor of where we are going now. And so what you're going to get in this next bit is a little bit of history that will end up in sort of new unpublished things to give you an idea of the kind of direction. And for the students that are here, I think what I'm hoping to get across is sort of the process of discovery and how some of these findings came about and how one thing led to the other. So these are the 3 themes that have really gone on in my lab for the last many years, trying to ultimately understand what makes a stem cell and how does it go bad. And it's important, and as I said, it's going to be hard to do this in a short period of time because we've done actually a lot of work. The students in my lab have done a lot of work. So I think it's good to start here because, for those of you who don't know, I mean, the era of modern stem cell research began in 1962 with this amazing paper that Till and McCullough reported on, and they were not, looking for stem cells. They were, this is 1962, this is an era where radiation was really thriving in cancer therapy. remember, I mean, the atomic bomb only was, dropped a little bit before that time, and the whole atomic era only began a few decades before that. And so people were very interested in just understanding how do cells respond to radiation. And one of the experiments that they were trying to do was to understand how bone marrow cells respond to radiation, because it was going to be a prelude, both looking at toxicity if you irradiate some tumor or some tissue, but you need to understand how bone marrow works, and perhaps you could have therapeutics along this line. And so what they did is they took bone marrow, gave different doses of radiation, and for a couple of reasons they decided to look in vivo. And they would look 14 days later. So these are mice that were irradiated. The mice are going to die in about 8 or 9 days. And it was actually known from the 1940s, because when people were just looking at what does radiation do, they knew that that the most sensitive tissue in the body was the bone marrow, because it's the most replicative. And they knew that they could rescue that by doing a transplant. So that was known in the decades before. And so they were just using that tool as a way to just understand how radiation is killing cells. And so they gave these graded doses. And they looked on the spleens of these mice. They looked at different tissues, but they happened to look on the spleens. And what they realized was there were nodules on the spleen, only of the mice that got cells. If you didn't get cells, you just had a tiny little spleen with nothing on it. But the insight was that the number of nodules on the spleen were related to the dose, inversely related to the dose of radiation. And when they plotted it, it was a straight line. So, and they knew that, radiation, photons, or the radioactive particles, are discrete entities, and so they hit with single hit kinetics, and it was a straight line, meaning that there was a direct relationship between the cells that are surviving radiation and the single input. And then they went on to prove that, you open up that nodule, there's about 1 million cells, and it was all multiple lineages of blood, and that means that one cell made all multiple lineages of blood. The textbooks before said that there would be a stem cell for each of these lineages. 1940s, you see stem cells, but stem cells for each lineage of blood. And what they proved was that a single cell actually makes multi-lineage outputs. And so that was the birth of, and then they made a bunch of other principles. And so what that led to was the idea that there is, this hierarchy. Others worked out, they actually contributed to this, that, you had these transplantation assays that had these multi-lineage stem cells, and others developed colony assays in gelatin and putting in a lot of goodies, and you could see these colonies forming, these clonogenic cells, but the clonogenic cells often were unilineage. And they put all these things together, and because they were counting, right? All the colony assays were made by they were measuring single cells, and they were counting stem cells all along the way. They were very quantitative, and that's what they could then draw a relationship between one cell and build the whole tree. And so that's what we knew. And so when I started my lab I had worked on the mouse system. I'd worked on mouse stem cells, putting genes into mouse stem cells. This was the era of retroviral vectors had just been discovered. Alan's lab was the first lab to put retroviral vectors into actually these colony-forming cells, and into the clonogenic cells. And my postdoc project was to try to put genes into these colony-forming cells in the spleen and the long-term repopulating cells, because others had realized that if you put a cell in a mouse and let that mouse go for a few months, you actually can read out a cell which is probably earlier than this CFUS. I stayed in Toronto, I got a job at Sick Kids, and I wanted to differentiate myself from Alan, who was across the street. And so I decided I wanted to work on the human system. And we wanted to put genes into human cells. And so we had colony assays that many people had developed in the prior years, and so we put genes into them, and it was very efficient. But I knew from my mouse work that colony-forming cells aren't stem cells. And so the question is, what's the stem cell assay I'm going to use for a human? And of course, the guiding principle is that we can only assay stem cells based on transplantation. Nobody wants to volunteer for my experiments. So what are we going to do? Well, it turns out that one of my colleagues, my mentors, Bob Phillips, was the world's expert in immune-deficient mice. And these are mice that had a natural mutation in the SCID gene that is involved in recombination on T cells and B cells. And these mice don't have T cells and B cells. And so the thought was that if we put human cells in, they're not going to be recognized as being foreign, and therefore they might engraft. Maybe there's enough goodies between what a mouse can provide and what a human cell needs. And it turns out that that works. And so now we had a mouse running around, and we did what a stem cell biologist would do is we measured it, we did quantitative analysis. Some mice got 10 million cells, some mice got 5, 1, and so forth, and we showed that the frequency of repopulating cells was about 1 in a million. So this is, trying to find what is the ultimate human stem cell was searching for a needle in a haystack. But at least we have an assay system. And so we spent a lot of time in those early years just to figure out how can we make this assay better, more efficient, and so forth. We came up with different strains of mice, but ultimately, it would set off a search that took us almost 25 years to get to the next step. So what was known by us and others was that there was a set of markers that CD34 and a few others that one could, oops, just a second, back, there we go. I guess I can use my pointer. Yep, there we go. So what the Weissman lab had shown was that there was a new marker, CD90 or Thi1, which was expressed on what they had an assay for a stem cell, is a bit different than ours, and that seemed to distinguish the long-term and the short-term, or these two different stem cells. We looked at their data. Actually, Fayez Nada was a green PhD student with Sergei Dulatov, and they looked at their data and they realized that they had missed, or they hadn't talked about it, but actually there were still stem cells in the Thi negative cells. There was a higher frequency in the Thi positive cells up here, but it was, there still were cells there. And so the question then was, well, what do those cells look like? Thi isn't enough. And there had been work in the mouse system that had suggested that integrins would be distributed or expressed differently across the top end of the mouse stem cell system. So we looked at human integrins in this set, and we measured them with antibodies, and we realized that integrin alpha 6 or CD499F was expressed at different levels in the different fractions. And interestingly, we found that there were 49F positive cells in the Thi negative or positive population, and they're also there in this negative. And so we sorted based on CD499F, and then did this, we showed that we got engraftment, then we did this, the definitive experiment, which was we did sorting. So this is like at the time, this was like really complicated, still complicated sorting, but with the instruments we had 5 years ago, this was a pretty challenging experiment. So we did sorting, we single cell dropped the cells in wells, and we visualized that there's a cell, sucked it up with a syringe in a needle, and then we injected it in the mouse, single cell. And so that way we could ask if mice were engrafted, then we knew how many single cells or how many stem cells there were. And it turned out that there were about a third to an eighth of the mice were actually engrafted, meaning that the purity that we had achieved from 1 in a million, we now had 1 in 3 cells in that pot were actual stem cells, so almost purity. And these cells met all the criteria because they made both B cells and T cells, myeloid cells and lymphocytes, pointing out that these are multi-lineage regeneration. So this was a real important advance. We'd gone from the discovery of an assay to the actual discovery of the human stem cell. And of course, this coincided right around the time when technology became available to do all kinds of single cell analyses and omics that I'll describe in a second. And we did a lot of work, a number of other postdocs worked on trying to actually build an idea, the 49F positive cells, the 49F negative cells, then the 90 negative cells, and we could show that this was actually a hierarchy. And the interesting thing is, if you transplant all these cells, they're all multilineage. So multilineage is not a criteria for being a stem cell. It's just that the longevity of the graft is different. These only do it for weeks, these do it for months, and these ones will do it longer. But the key is, the hallmark property of a stem cell is not that it makes progeny, but that it renews itself. So when it divides, it actually replaces itself, and that's why stem cells can be, have longevity. In a mouse system, a single stem cell can regenerate a single mouse for its lifetime, and you could actually do it again in another lifetime of another mouse. So it's a remarkable output of a single cell. In a human, we don't know exactly what the output is, but it's a massive output in a human. And so, we now have a better hierarchy. I'm going to just summarize, actually, this is our COVID project of the lab. I mean, of course, it started earlier, but these 4 papers here, this 5th one was done earlier, but these 4 papers here were reported in the last 3 years. And they basically have given us, by looking at the long-term stem cell and the short-term stem cell, one cell has self-renewal, one cell doesn't. one cell is very dormant, the other one isn't cycling, but it's a little more activated. And that has given us a view of, at single cell resolution epigenetic analysis, we know that there are 280 regions of the genome that are open in that long-term dormant cell. They have CTCF bound on one site, and that has to close down and repress when the cell gets activated. And that is a beautiful signature of dormancy. And that signature turns out to work not just in blood stem cells, it works in other kinds of stem cells as well. And the other thing we discovered by complete serendipity was that many cells in the body, when they get stressed with DNA damage, protein folding, and so forth, they pause and they resolve. They put up chaperones and things like that to try to resolve the stress, and then they go on. If it stays on too long, then eventually the unfolded protein response starts and the cells kill themselves. But what happens with stem cells is that they're tuned exactly the other way. If they get stressed, they get culled right away because the PERK pathway is very high in stem cells compared to the cells downstream. And the idea, evolutionary logic, is that these cells need to preserve themselves with any kind of damage. There's a possibility of having damage in a stem cell which will be propagated to potentially trillions and trillions of progeny over the course of decades that now sets up, every of those cells will have that single bit of damage. Then you get second damage and so on and so forth, and the prospect of malignancy. So stress regulation is different in these cells, and metabolism is different in these cells. I'm not going into details. And the paper at the top here, we've actually uncovered that, we often think dormant cells are just quiet cells, they're not cycling. In fact, dormancy is a programmed state. And one of the aspects of the program is there's very high endolysosomal activity. The cells have very active lysosomes that are always cleaving receptors off the surface of the cell, meaning that even if there's a signaling molecule that comes on, if it's being cleaved off, it can't signal this dormant cell. So it's like a physiological way to maintain dormancy. And then the other new property that, again, came just, as a cell and a gene that came from a screen is a gene that we uncovered called INCA1. It was only 3 papers in the literature when Kirsten had looked at this. It seemed to cause an increase in stem cell activity, and we wanted to figure out how it worked. And so what that led to was the idea that, yes, we have this long-term stem cell, and I kind of have alluded to the idea that this is a homogeneous cell, right? This is a pure population, very identical all the cells in that population are always doing the same thing. In fact, what we've uncovered is that there's heterogeneity in that. There's a population of cells that do 2 different things. And the 2 different things that they do is that when they get transplanted, one population, the green population, gets going quickly and contributes to the graft because this mouse has demand. If this was a human, this human has demand to get the stem cells active. But that comes at a cost. And the cost is that stem cells are lost in the process of regeneration. Whereas there's another population of cells, the red population, which takes a while to get going. It doesn't contribute early on, but later on it does. And what this is a mechanism where the stem cell are preserved. So these are the ones which are going to contribute to the engraftment of this person or this mouse long term. And this data is actually very consistent with, so this was done on a mouse system, human cells in a mouse. But the data is very consistent with human gene therapy trials, where you can take the retroviral insertion sites, many hundreds of thousands that a person gets when they get a gene therapy, and the group in Milan has tracked these over the course of many years, and they can see exactly these kinetics. And so we think that this explanation and this mechanism is underlying that. The details, again, I can't go into, but essentially there's an epigenetic player. There's H4, K16 acetylation marks that are ultimately governed between the dormant state and this activated state with this molecule, INCA1 and PEC4. And so we think that latency is a hallmark of the most primitive cell. The fact that it doesn't get activated when you put demand on it right away. And so, to get more understanding of mechanism, we, like others, are undertaking, the single-cell omics, single-cell RNA sequencing, and we all have seen this great papers, nice clustering, you do UMAP projections, and you put labels on them, and it all looks well and fine. And the problem is that all the data around the bone marrow was done on bulk cells. And I already told you, even if you purify with CD34, it's still a relatively small number of cells, and the number of long-term hematopoietic stem cells is, it's like 1 in 1,000. Like there's probably 1 or 2 dots in any of these things that are actually true long-term stem cells, even though I have a label, HSC. So we needed a better, map of human development. And Andy Zhang, who's an MD PhD in the lab, sort of took all the data that was available and through a number of computational approaches was able to get a much more balanced map where there are many more long-term hematopoietic stem cells that are represented in this map. And so we now have a very finely graded view of how an HSC here develops into lymphocytes on this angle and erythroid cells and so forth on that angle. And so just to give you a flavor of how good this map is, we have our own data that we've generated on these highly, highly purified cells. And we know that a portion of these cells we put into mice, and we know there's repopulating cells there, and we've done RNA sequencing on them, and we ask, well, where do they land? And they always land in this map that we call, HSC. And then here's the best one, which is, this is 5,000 purified long-term HSCs, and they all land in this window. And then there's a few other papers that were reported. So the point is, because we have this independent validation, we can now have a lot of confidence that when we have a cell, an unknown experiment, and cells land in this window, we can have confidence that these are actually the stem cells. And so when we started to look at that and investigate it in more detail, Andy looked at this data and realized that this population is not homogeneous. In fact, we see that there are different kinds of pathways or cells that are activated in different kinds of pathways. And a lot of them is this sort of ER stress, I already talked to you about that, but inflammatory programs. And if you look at that nest of cells down here, you can see it's very much guided by classical definitive immune response signatures guided by NF-kappa B. Here's the ER stress pathway. That's why you can label it this particular way. And so this got us thinking about, well, what is it about inflammation that is underpinning this subset of dormant long-term hematopoietic stem cells? And so this is a project this is the new part. So Andy Zhang is still working on it by a new postdoc, Murtaza Nagri, and guided by Stephanie, who's an affiliate scientist in the program, who's really driving this subset in the lab. And so the idea is that, we know in the blood system that there are a lot of tissues that respond to inflammation. And so what we know is that T cells and molecules like that will respond. They get activated, and in some cases those T cells will revert and create a memory T cell. Right? I mean, the idea that we're a child, we get our immunized, and we make B cells, we make helper cells and so forth, and then we don't see that antigen for 30 years, but we see it 30 years later, and these memory cells can respond. So we know that, that how they exist. So, that's kind of well studied. But, what about at the blood cells, at the stem cell system? So there's beautiful work in the mouse that has shown that if you do an acute injury, you basically can really affect stem cells very quickly. They differentiate. And if you do it chronically, what happens is you really deplete the stem cell compartment and you really enhance myelopoiesis or myeloid fate. And that's actually what happens in aging as well. So as mice age or as humans age, this is what you get naturally in an aging blood system. So what's the link between aging, chronic inflammation, and then of course, more and more work is showing that if this goes on too long, you start to get alterations that really sort of lock this in, and that is essentially the beginning steps of malignancy. And so we carried out this experiment. We took cord blood cells, a very naive population, put them into mice, waited a couple of weeks for engraftment, and then we treated with one dose of TNF, one dose with LPS, and you can see it looked 16 hours later, massive acute response, just what we expect. And then we wanted to look at recovery. 4 weeks later, another 2 weeks later, and you can see the cells have almost recovered, no matter the treatment. But the kinetics at these different time points are quite different of the stem cells. Stem cells are dormant here. By this time, they're all cycling. And then if you look 20 weeks later, most of these cells are actually quiescent. And when you look at these mice 20 weeks later, you can see that there's actually been a permanent impact on the hematopoietic system of mice that got a single exposure, or in this case, 2 exposures of these inflammatory agents. And the question is, what's guiding that? And so we undertook, in the latest, greatest sort of omic technology now, which is on single cells, we can take the cell and you can get the nucleus, get the cytoplasm, you can do RNA sequencing and ATAC and get an understanding. And that's really good when cells are very similar. So it's very hard to find RNA differences, so gene expression differences if cells are very similar. But chromatin accessibility is a very good, better way to predict state. And by doing a joint embedding, what we realized is there's 2 stem cell populations. There's a stem cell that we've studied before, we reported on. It's connected to the rest of the blood system. But there was another population off to the side, which was very unusual. We knew it was a stem cell based on these signatures, but it had other gene expression features to it, and actually also epigenetic features that made it very distinct. And so the question is, well, what is it? And so we compared how it relates either in a control setting or after TNF treatment. And what came out of that was that this HSC2, weren't very imaginative, so the new population was much more different than the other population in terms of gene expression. It changed with TNF. Same thing true with ATAC. There are many more peaks coming up in this population. And of course, this data can be brought together and we can really understand transcription factor binding sites. And you can basically see that TNF is not touching this homeostatic population, but it's really touching this HSC2 population. And if you look at the players here, quite a few of them are around, anti-kappa B and so forth. So these cells are somehow responding and have some memory because the TNF exposure was done 2.5 months earlier. And so this just summarizes that data, and essentially what we think we've uncovered is a brand new stem cell population, which in itself is a biosensor for inflammatory response. And it appears that this is, a cell which is the cell which is responding to this inflammatory insult and has a memory, retains a memory of it. Memory is presumably encoded. We have to investigate the epigenetics more, but it's encoded in the epigenetics. And so the question is, well, how is this related to aging? And so we have access to data in our own lab where we have an aging project where we have bone marrow from patients in their 20s, their 50s, and their 70s. So we created a signature of this inflammatory HSC inflammatory memory. And you can see it's very low in cord blood. It's still quite low, but going up in the young bone marrow, but really goes up in the 50-year-olds and stays up, perhaps goes down a little in the 70s. We can talk about why it might go down. And then there's other independent data sets that's actually been published, again, young and old, increase in signature, pediatric and adult, increase in signature. So it looks like there's a relationship between this new stem cell population and the natural process of human aging. And then, I told you chronic inflammation is linked to malignancy. And so we had access to a brand new paper that was published. This is an engineered leukemia model where they take normal, actually iPSCs, and they take the iPSCs and they knock genes into them, and then they drive them into blood development. And so they can drive them into blood stem cells. And so they look at the blood stem cells from the normal cells, the ones that have 1 mutation, 2 mutations, and 3 mutations. And this mimics the pre-leukemia progression. And when you look at the signatures that are there, the signatures in these early pre-leukemic components are actually the same signatures that we see in our inflammatory memory signature, FOS, AP1, JUN, so forth, that are really strong inflammatory components. And so we think that what we've uncovered potentially is the source of a cell that responds to chronic inflammation, but one that's also progresses towards malignancy. And so we're really going to explore that in more detail. Okay, I'm going to just move through this last bit a little bit more quickly because I'm going to be running out of time in about 6 minutes. So Gabby already sort of pointed to this. If I often say this in talks, if I ask you what's cancer, or I stand on the street corner and ask somebody walking by what's cancer, I'll generally get the same answer. Right? Cancer cells that grow in an uncontrolled way. Cancer cells that grow too much. And that concept has undergirded cancer therapy for 8 decades and driven the development in the early era of anti-proliferative chemotherapy drugs. Right? All our broad spectrum chemotherapy drugs are killing cells that grow too much. And that's fine, but if you ask the question in a different way, and that is, is every cell in a cancer equal? And does every cell in a cancer have the ability to keep that cancer going? Because remember, tumors are dynamic entities, just like our tissues are, right? Cells are being born and cells are dying. Tumors, cells are being born and cells are dying. So if you have a cell that's, born today and it's dying a week from now, that cell's not very important to me. What's important for me is the cell which is going to keep that cancer going for the next 10 years or 5 years and maybe survive therapy and going to kill me because it's causing relapse, right? So we need to understand the contextual property of these cells. If every cell is equal, we can have one kind of therapy, but if other kinds of cells, only some cells keep the cancer going, then we probably want to target those cells and try to understand them. And the problem, the paradox is that leukemia cells can be running rampant in the bloodstream of a patient, but we can't study them very well in a human because we can't put them in a lab dish. And that's true 50 years ago, and it's actually still true today. And so people in the past era would have reverted to mouse models of the human cancer, and we have we've made many advances from that. But at some level, we actually want to study the human disease, right? A mouse makes as much blood in its lifetime as a human does in a day. So, the output and the demand is quite different. So we need to understand that. And so because we've already put normal human cells into mice, we thought, well, let's put leukemia cells into mice. And turns out that works. So they don't grow in a lab dish, but they all grow in a mouse. Mouse running around, we did the same classical experiments, asked how many cells do this? It's 1 cell in a million. Now that was a quiz for us, or an issue, because of course, there had been work that McCullough and others had done decades before we started these experiments that had done the same kind of colony assays, and they showed that only 1 in 100 cells can make a colony in a dish. And they did a lot of work on that clonogenic assay because that was the cell that you needed to target with chemotherapy and so forth. And so the question was, our frequency was 1 in a million. So we have 2 options. One is it's an ancestral cell, earlier than a colony-forming cell, just like my trees showed earlier, stem cells and the colony-forming cells are downstream. Or it could mean this is just a really crappy way to study, clonogenic cells. This is just the clonogenic cell, put it in a mouse, and only 1 in a million actually read out. So we had to figure out a way to sort the cells that had clonogenic activity away from the cells that had stem cell activity based on repopulation. And so this is what the work of C. Lapido, who's my second postdoc, did this work. And we basically took AML cells using some cell surface markers that were differentially expressed. We sorted them into these different pots, and we transplanted these different pots into mice. And the pot I'm just pointing out down here represents only 1% of the tumor, right? 99% of the tumor were in the other 4 quadrants. And whenever we transplanted these into mice, the question is, where does leukemia come from? And I should just tell you that the R1 there is where a lot of the clonogenic cells were, right? So that's where all these colony-forming cells were. And we put them into mice, and the data was very clear. This population up here never gave engraftment. This is the level of engraftment. Each dot is a mouse. No engraftment, no matter how many cells we transplanted. What is this, 5 million cells, never got leukemia. We transplanted the 1% population down here, and we always got leukemia across some graded dose. And so this was the definitive experiment that colony-forming cells found here can never engraft a mouse. Therefore, the cell that engrafts the mouse must be a different population. And that was the underpinning then of the idea that not every cell is equal. Some cells have stem-like potential, just like the blood system. And that concept turns out to work, as Gabby mentioned, across not just leukemia, but other solid tumors. We collaborated with Peter Dirks, who was a neurosurgeon, new faculty at Sick Kids when we were there, who took these concepts into the brain and reported the very first study of brain cancer stem cells in glioblastoma. Mike Clark, at time in 2002. Peter did the work at the same time in breast cancer, showed the same thing. Catherine O'Brien did colon. Actually, in this story, a lot of ovarian, pancreatic, lung, all these were done. The first examples of these were done actually in Canada. So this is Canadian contribution to cancer stem cell science that emanated from this work. And so what this led to then was the idea that tumors are caricatures of normal development. And if that's true, it means that the leukemia stem cells at the top are the only cell that's important to that tumor because it represents the unit of selection for that tumor. And so the question then becomes, challenge to us was, well, this is all well and fine in a mouse, human cells in a mouse, pretty artificial. Is this relevant in people, right? There's lots of things that happen in a mouse that have never translated very well into humans. And so we had to figure out how can we show relevance. And we thought, well, maybe the best way to show relevance is to try to identify the gene expression programs of leukemia stem cells and show that they have real clinical relevance. And so then the question is, well, what's the clinical relevance that we could use? And so I thought, well, clinical relevance in cancer is, do I survive my therapy or not? So the ultimate binary outcome, right? Am I dead or alive? So there's something that leukemia stem cells have that can predict whether you're dead or alive. So we did this complicated experiment. It took many years to do this. took almost 100 samples of AML, sorted it into fractions like that, subjected each of those to gene expression, but at the same time we took a portion of the cells, injected them into mice, and asked where are the leukemia stem cells and where aren't they, because it turns out what I showed you earlier works for some patients, but not every. Each patient is actually its own unique cancer, and sometimes the stem cells are in different places. Sometimes here and here, here and here, and so forth. And so you actually needed to do this complicated assay. But at the end of the day, we had all the fractions that didn't have leukemia stem cells, all the fractions that did, came up with a differential gene list, took the 104 top genes, and then we ran it against clinical data, where gene expression on 500 patients was linked to survival outcomes. And what that data showed was that if you have stemness genes, you can predict survival. We then did machine learning to refine this into a 17 gene signature. Each gene has a weight, some positive and negative, but you score that up and it turns out it's very predictive. We've worked with our molecular diagnostics lab to actually make this a clinical test. Very rapid, it's a nanostring, so you just have these 17 genes with some control genes, and we can get data back to them in 24 to 48 hours, and clinicians can make decisions. Because it turns out if you have a high score, you do much more poorly than if you have a low score. This is to come to standard chemotherapy, but the question is, what kind of drugs can I, it means that the drugs that I'm giving these people are not going to work on these, and I need to give these people something else. And so that's how we establish clinical relevance, but also how we're going to go on in the future. So I think what I'm going to do is, I'm going to skip over some slides because I actually decided to just talk a little bit more in detail about the normal stuff. And so you're going to see me go through a few slides because I want to just get to the end. Because I'm going to just skip over this. This is just some recent work that we've reported on. And just end up with the last part. And that is, we've also been very interested to ask, a person comes into the clinic and, gene sequencing is done in AML. They have 3 or 4 mutations. But the question is, what happened the week before, the month before, the decade before that person getting into the clinic? When did the cancer start? What cell did it start in? What was the first hit? Why did the first hit cause this pre-leukemia? What was the progression going forwards? And so we really needed a way to understand this in primary human cells. Apologies, I'm just going to skip over this. So this is reporting on 5 papers that we reported on in the last few years that give us a huge understanding of the origins. But what we really wanted to do was model this in a reliable way. And one of the diseases that we were very interested in is pediatric disease. And what became clear a number of years ago from twin studies, where one twin would get leukemia at age 2, often the other twin, if it was identical, at age 10 would get a very similar leukemia. And it turned out they shared a common translocation. And the only time those 2 babies were together was in utero. And what that led Mel Grease to recognize is that all pediatric leukemia starts in utero. The first hits happen in the fetus. And so how are we going to model, the data I showed you earlier was done on adult disease, but how are we going to model children's leukemia? Because these early steps, going from normal to pre-leukemia, happen, in the baby. And so we collaborated we focused on Down syndrome leukemia. And this is a very characteristic pre-leukemic phase. So children have trisomy 21. 30% of those kids actually are born with a pre-leukemia. It's a leukemia. It lasts for a few months, and it actually dies off naturally. Needs sometimes a little bit of chemotherapy. 25% of those kids will later on go on and get a full-blown nasty leukemia. And we know what some of the genes are, but we don't know anything else about this. And so we got together with people at Sinai, got access to human fetal liver from either normal people or trisomy 21 fetal livers. And then we engineered with CRISPR these different hits, GATA1 and STAG2, into different populations, and then we transplanted into mice. And everything is this is done by Alvin Wagenblast and Eric Leckman. And this is the summary of that data. And essentially what we showed is that we only ever got the progression from normal to pre-leukemia if we used trisomy 21. Never worked with normal cells. The second is, when we looked at the various populations, only the long-term stem cells, the most rare population, if we engineer into that, then we get pre-leukemia. If we use any other, even a short-term stem cell, we don't get pre-leukemia. So this is, we know something now about the cell of origin. We know something about the first hits. It must be something to do in trisomy 21. And there's some microRNAs that Gabby helped work on. And so these are the molecules that are really important in that. And then the second hits can happen anywhere. And so we learned something about the progression. So I just want to finish off by saying that, I stand up, I get these awards, But it's people like Gabby and this team and plus, the other about 130 or so people who have been through the lab, plus a whole bunch of co-op students. They're the ones who have not just contributed their hands to do these experiments. I sit in front of a computer, right? I don't do experiments. But it's the talking, right? It's the crucible. We all throw our ideas into this pot. And, I don't know an answer. They don't know what the results, what the results mean. But we talk about it. And what rises out, is the crucible of the new idea. And that's what they've really contributed to this. And they're the ones who I dedicate this to. Thanks very much. And of course, my family, who I wouldn't be here at all without them. Thanks. And I did talk long, sorry.
Okay, so we have some time, not very much. We've got a group of guests coming in to listen to Dr. Karikó in a few minutes, but we do have time for a few questions. And then at the end, after Dr. Karikó's talk and a short Q&A with her, people who want to stay for an informal discussion with both Dr. Dick and Dr. Karikó can do that. So if you have a question, there's microphones on either side. Please use the microphones because this is being recorded and we can't pick up the sound unless you use the microphone. So if we just do have a question or two, go ahead. Terry?
Hi, thank you, John. Beautiful talk, as always. Just a question, now that you have these signatures and they kind of look and feel the same between the normal and the leukemic, can you talk a little bit about therapeutics and how you can find something that's selective?
Yeah. I think that's the challenge, actually, to find a therapeutic window where you're not targeting normal cells and obviously taking out the leukemia cells. I mean, the thing is, the devil becomes in the details. And so actually there are differences, right? I mean, a leukemia stem cell, there's a lot shared, but there are some differences. And so that's the challenge. And I think we have these single cell tools that we have actually give us that insight that we can then find the sweet spot. Sometimes there could be a cell surface molecule on a particular leukemia that's not on the normal. Maybe that's a target for a CTL. There's some molecular pathways that might be slightly distinct. So there are tools that I think that are available. There are insights, let's put it that way, that we've begun to mine going forward. So.
I always have a question. No, it just got me thinking because I heard your talk yesterday too, John. The HSC 1s and 2s, is there crosstalk between them? So under stress conditions, does the HSC 2s affect the HSC 1s?
I mean, we don't know that. This is a recent discovery. This is just the last month or two. So there's a lot we don't know. We don't know really what the relationship is between the two, how much one makes the other. So these are ones that, these are important questions, let's put it that way. But they're ones that we have traction to get at now.
Okay, so maybe we will stop here. There will be a chance at the end, as I say, for people, just a general discussion. We've left time for that. And we'll take a very quick sort of break, no time to go anywhere, while people get settled. And then Gabriela will introduce Dr. Karikó.
Okay, we would like to acknowledge the presence of Her Excellencies, the President of Hungary, Katalin, who is here to listen to Dr. Karikó's lecture. It is now my pleasure to introduce our second speaker, Dr. Katalin Karikó, a pioneer and an advocate for the field of RNA biology. Dr. Karikó's research has been in the development of in vitro transcribed mRNA for protein therapies, and her work has laid the groundwork for the mRNA vaccine, including the COVID-19 vaccine made by Pfizer, BioNTech, and Moderna. Dr. Karikó's inspiring story really exemplifies that hard work, perseverance, and willingness to accept failures and keep trying is a key to scientific discovery. Dr. Karikó was born in Hungary, where she earned her PhD in biochemistry at the University of Szeged and worked as a postdoctoral fellow at the Biological Research Center of the Hungarian Academy of Sciences. She then moved with her family to Philadelphia for a job as a postdoctoral student at Temple University, where she now serves as an adjunct professor. Dr. Karikó is a senior vice president at BioNTech RNA Pharmaceuticals and has earned countless national and international distinctions. Dr. Karikó, alongside with her collaborators, Dr. Weissman at the University of Pennsylvania and Dr. Cullis at the University of British Columbia in Canada, have been distinguished this year for their pioneering work in developing nucleoside-modified mRNA and lipid nanoparticles drug delivery, the foundational technology for the highly effective COVID-19 mRNA vaccine. Dr. Karikó and Dr. Weissman overcame challenges associated with mRNA delivery by modifying the RNA so that it could resist quick breakdown and avoid activating RNA sensors. This mRNA modification, together with the lipid nanoparticles technology developed by Dr. Cullis, enabled rapid production of the highly effective and safe COVID-19 mRNA vaccine. Their combined discoveries have translated into an important tool for the control of the COVID-19 pandemic and have the potential to revolutionize the future delivery of effective and safe RNA vaccines, therapeutics, and gene therapies. Please join me in welcoming Dr. Katalin Karikó to the stage. The title of her talk will be Developing mRNA for Therapy.
Thank you very much for this nice introduction. I am first time at York University, first time in Toronto. I have been in Canada before, but not that part. It's a very beautiful, large city. So today I am here to talk about developing mRNA for therapy. Due to this vaccine development, most of you already know what mRNA is, but I will talk about how it was developed for therapy. So messenger RNA was discovered 61 years ago, and it took 60 years to develop it in an FDA-approved mRNA product in the form of COVID-19 mRNA vaccine. What happened during these 60 years? There are some major things, but what I could say that most of the time just the incremental increase, improvement of the RNA, the purification, the delivery, this is what happened. So I will talk about these years. I have to say that in 1961 I did not read the literature yet. Maybe I couldn't even read Hungarian, but I most of the time, I was on this field, and I remember. So for me to go back and talk about the history is much easier. So the messenger RNA was discovered in 1961. This was these two papers reporting this labile intermediate. And then with this one, actually, I don't have time to talk about those 50s when they try to find this molecule, but with this one we learned that what is the information flow is in every cell in our body, and actually every free living organism, is that from the DNA there is an RNA is made, and this RNA, messenger RNA, is translated in the ribosome, and the protein is produced there. So at the beginning, scientists, they couldn't synthesize RNA, so we couldn't make anything like the vaccine. What they could do is they isolated RNA, and they started to investigate it. First of all, it was discovered in the 72 already that the RNA has a poly A tail, and in 1975 they discovered the 5' end had this cap structure. And scientist Furuichi was the first one who reported. I was lucky to meet him in Japan when I received the Japan Prize. Unfortunately, 2 weeks ago he passed away, but he was the first one who promoted and suggested that this is the 5' and how the 5' and the mRNA looks. And if you get the Pfizer vaccine, that was incorporated, this kind of structure, Cap1, during the synthesis. And during this 1975, several papers published on how the cap looks. And in 1975, you work something in the cap. Probably you could get a Nature paper easily because you can see that all of this paper was published on that year. By the end of the year, Bernard Moss and colleagues discovered that actually there are viral enzymes which can put this cap on the RNA, and then if you get the Moderna vaccine, these enzymes were used, actually, vaccinia viral enzymes were used to cap the RNA. So these scientists were analyzing RNA on a way that what structural elements are there. Other scientists started to look at that, how RNA might be translatable. They again isolated RNA. These were from reticulocytes, which is the precursor of red blood cells, and they could purify this globin RNA fraction, and they translated in cell lysate, or in the 60s, or they injected to frog oocyte. They were large cells, and it was easy with a, I am saying easy, I have seen people, it's not easy to inject with a glass capillary. Or actually in 1978, when first time RNA, this isolated messenger RNA, was delivered to primary cells using liposome. And this Dimitriadis did this actually, and Ostro. Dimitriadis worked in London, and this is where the first time liposome was developed. And what happened is that putting the RNA into this liposome, this isolated RNA, and they fused to the lymphocytes, which normally would not express globin, and they could show in this Western blot that the protein was made. So this was the first time really shown that mammalian cells can translate an RNA which was delivered to the cell. And prior to that, other scientists actually used already liposome, delivering plasmid to the cells. And in Hungary, where I work in the lipid team, we also were interested to deliver with liposome nucleic acid to the cell. And my advisor was Ernő Duda and Eva Kondorosi, as I just learned that Ernő Duda did a 1-year sabbatical right here at York University. And so what we have done, we couldn't purchase this phospholipid fraction. We were behind the Iron Curtain, so we just get the cow brain, because that's what describes how to isolate phospholipid. At that point, came handy that my father was butcher, so it was not gooish working with a cow brain. And we, after 1 week work, we isolated this phospholipid, phosphatidyl ethanolamine, phosphatidyl serine fraction, and we did a liposome, and we delivered plasmid to the cells. And this is for the undergraduate, because I was undergraduate student, so it is important to make your hands wet. Start with some experiment, and maybe rest of your life is thinking about that. That's my advice. From the lipid team, which I get there by chance, and everybody thought that lipid is very boring, I accidentally went to the RNA lab. At that time, I didn't know that maybe one day this lipid and RNA will be so well composed in a vaccine. Here, my supervisor was Jenő Thomas, who actually made cap analogs for Furuichi and Aaron Shatkin and sent it to New Jersey, because when you analyze something and identify something, you need a reference material. Somebody would be, organic chemist would synthesize the molecule, and then you can compare. So 78, when I started my program there, in PhD program, I already knew day one that the mRNA has a cap. But my role was different. I had to chemically synthesize 2', 5' oligoadenylate. These are antiviral molecules. It was just discovered the prior year by Ian Kerr in London. I worked closely with János Ludwig, again, I am saying that we were graduate students together, and even today I ask organic chemists questions from him. So it is good that if you nice to your fellow graduate students and follow them what they are doing, so one day you need advice, they will respond your email. So we work on this 2', 5' molecules, and actually this naturally oligoadenylate synthase is synthesizing in our body in response to double-stranded RNA when there is a viral infection occurring. I have to say, again, not just what your colleagues are doing follow, but follow also what you started. I did this experiment in the 70s, but I have to say that oligoadenylate synthase is one of the 3 genes which polymorphism promoter can cause somebody that when infected with coronavirus, they get seriously ill. So this OAS actually synthesizing in our body this short RNA molecule, which activates an RNase L, the latent enzyme, cuts up the RNA, and that's what first we thought that this is the basis of antiviral effect of the interferon. And as you know that everything I am saying RNA, because we synthesize the RNA, we have to make a template also or substrate for the RNase L, so that it was always working with RNA. But we couldn't deliver very well, and so it was the company in Hungary, pharmaceutical company, did not support further our studies, so I had to leave and come here or to the United States. And I might mention here, I wouldn't stand here if I would not be terminated in my position several times. So it is important that what happens to you, what decision you make after you terminated, and that's why I am here. So I went to Temple University, and we were working on the same molecule. We tried to develop an antiviral compound, but when there we needed very much an antiviral compound, I am talking about 1986, the major problem was HIV. And we couldn't deliver this molecule, again, here at Temple University in the US. So eventually we treated HIV patient in 1986 with a mismatched double-stranded RNA, which called Ampligen. In 2001, actually, we learned how double-stranded RNA can activate interferon and induce interferon when toll-like receptor 3 was discovered. At that point, we just knew from 1963 that double-stranded RNA can induce interferon and it can help get antiviral effect. And unfortunately, HIV patient interferon system is compromised, so it didn't help them, and they died. But again, now we are working, I am working here with double-stranded RNA, so the interest towards RNA was always there. And while I was working at Temple University, scientists and Harvard University come up with the idea how to make RNA in a tube, so that without this, we wouldn't be able to make a vaccine RNA. So what they discovered is that the phage RNA polymerase, the first SP6 RNA polymerase they described, it is with high fidelity, it can generate from the template DNA, can generate the RNA. And you just have to add the basic building blocks of the RNA, and in our body also, every RNA what we have is from the 4 basic nucleotides is synthesized. And of course, Bernard Moss gave them some capping enzymes, so the RNA had all of the structural elements, the cap, the poly-A tail, and coded for interferon. And because we are in the 80s, so in the 80s still not much tissue culture experiments could be done, so they injected this in frog oocyte, and then cultured. And what happened in the culture medium, overnight culture medium, when they took, it had an antiviral effect. So this was the first time by design in ex vivo in a tube was synthesized an RNA, and this RNA was biologically functional. I might just mention here, you can, Douglas Melton and Paul Krieg were describing, and Maniatis and others also contributed. But when I sent in the 80s Paul Krieg a letter and asked him that, can you send me this plasmid? You know, he put in the envelope And hopefully, there was no empty, and hopefully nobody is in here from the university administration. So he just sent it, and he even put there the T7 and other plasmid. He said, we made a better one, and he sent both. So this was the 80s. Every colleague tried to have the other colleagues. And so that experiment inspired a lot of people, that we can make already RNA. And what you can see is then the 1990s, more and more paper came about RNA. Of course, you can see that every year just was one paper, so it was easy to follow the literature, not like in these days that, every day several papers. But it was crucial when John Wolf and colleagues showed that injecting an mRNA to the animal, they can detect the protein. For me, this paper was most important. Floyd Bloom, who was the editor of Science, he's a Scripps Clinic, he showed that an mRNA coding for vasopressin injected to the brain of the rat, it can cure a disease. Yeah? And then I wanted to make also, at that time I already was actually had a couple of grant was rejected in 1990. I already rejected grant on mRNA therapy, but this paper convinced me that it can be used. I just have to keep trying because they could do it. There were two papers at that early time tried to use messenger RNA for vaccine. These were both of them for influenza, and this French team they used the liposome to deliver. Why Lilstom, he just injected naked RNA. And if you try to look at whether these scientists ever published on RNA, don't look, because they did not. I followed all of the co-authors on that paper for decades and never published on RNA. And only these people who use for cancer vaccine, they did more studies and actually even started a clinical trial, Eli Gilboa and colleagues. So why do people stop using the RNA? I have to say that the RNA had only problem. Very small amount of protein, very short period of time, and it was when we showed also that it was inflammatory. So it has just problem, and nobody thought that it could be used for anything, so therapeutically, only just like a reagent in the laboratory. So what happened those 60 years, as I mentioned, RNA structurally and purification and many other ways were constantly improved, and together, parallel and sometimes common, the delivery system, because the RNA degraded immediately when injected to the body. So I was interested in RNA, and first I started using messenger RNA when I went to University of Pennsylvania in 1989, and Eliot Barnett, who was just 8 days younger than me, I convinced him that mRNA would be the way to develop this program. He was interested in urokinase receptors. And I have to say this protein, why I am talking about, because this protein is post-translationally heavily modified. There are glycosylated, there are GPI links. And interestingly, we put the RNA to the cell, and the cell, voila, knew what to do. It decorated properly, and the receptor was functional. So at that point, we thought that RNA, messenger RNA, will be good, if nothing else, for cell therapy. And we were thinking about bone marrow transplantation. We thought that we were delivering telomerase, and we will extend the lifespan of those cells. But here again, I had to leave, and I was demoted. And so I ended up in neurosurgery. That David Langer, who was actually a medical student in the prior lab, and he learned that I lost my job, and probably in the other lab I was kind to him, and he convinced the chairman that neurosurgery needs a molecular biologist, and this is the lab I got from neurosurgery, and I worked there 17 years. And sorry about no lab coat, no safety goggles, and no gloves. But we try to develop therapy for treating the stroke patient. I have to say that, you see that I was working with organic chemists when I have to set up an antiviral lab. I work with cardiologists. I work with a neurosurgeon who came from the seeing patient, and he had described the problem. And together we try to come up with solutions so that we educate each other and then innovate. So we want to actually develop nitric oxide synthase encoding mRNA for treating patients who subarachnoid hemorrhage, they get vasoconstriction. And he said that he can reach these major blood vessels, and the nitric oxide synthase would locally generate the NO, which will, millisecond, couple of millisecond half-life would dilate the blood vessel in the brain. So that was our plan. You don't look it up. I never published on it. We ran to Chicago. We were in different places where their model system were there, but the background was changing so much that we were not sure that in tissue culture we could show that an NO was generated. So we were doing this experiment, and we did inject it to the animals. I never realized that this mRNA was anything wrong. I met Drew Weissman, actually, as it described, is a copy machine. Again, it was 1997, 98. If it would be 2002, I downloaded every paper digitally. I never went to the Xerox machine. But that time I was still there and met him, and I bragged, and I told that I can do RNA. And he told me that he came from Fauci's lab, which I was not familiar with. He was not in the TV at that time every day. And he wanted to develop HIV vaccine. So he was interested that I will make mRNA for him, immediately offered. And he tested out. He said that he is using human dendritic cells. I have to say, again, in 97, I thought dendrite, probably some kind of neuronal subtype, but it is the immune cells, of course, we know very well. And he delivered the RNA with lipofectin, and then we published this paper in 2000 showing that mRNA is an excellent vaccine. It upregulated all of the markers, so much protein was made, and he was very happy. He measured that a lot of TNF-alpha was made. I mean, we just heard John's speech here talking about how TNF-alpha caused the pre-leukemic signature on the stem cells. So of course, TNF-alpha is last thing you want to deliver, or induce in a patient who's suffering stroke. So I was thinking that, 10 years I was working to develop therapeutic mRNA, and we cannot do it. So we were thinking that why it is inflammatory. I thought that the mRNA I am making is the same as in our cells. But we thought that maybe because it's coming from outside, and this is a sensitive immune cells, and recognizing that mRNA shouldn't be outside of the cell. So to test out that whether if we take out the RNA from mammalian cells and now exposed to these immune cells, whether it will be immunogenic. And this was the experiment, actually, which resulted to discovering that nucleoside modification is important. What happened is that we did made the RNA. This purple is one. You can see the most inflammatory. We measured the TNF-alpha. And different compartment, we isolated the RNA, and some of them was not as much inflammatory. That bacterial RNA was very inflammatory, but the tRNA was not inflammatory at all. Knowing that the tRNA has a lot of nucleoside modification, one idea was that could it be that this modification is suppressing immune activation? I just want to scare you a little bit with this one picture showing that how many different modifications the nucleosides can have in the mRNA. So there are more than 100. So we were thinking that how we could incorporate that to the RNA to prove that nucleoside modification suppress immunogenicity. And the enzyme even which incorporates this to our RNAs was not known, so we couldn't call Promega, send us this enzyme. Only 2 modification was in mammalian cells, which was well described, and this was actually my fellow University Szeged student. We lived in the same dormitory, we worked in the same biological center, and he discovered in the early 90s that how 2' methylation and pseudouridylation occurring in our ribosomal RNA. Yes, you can imagine that I called him up and asked him that to send those stuff. Tamás, send those stuff, I have to modify the RNA. But if you are familiar with the field, this requires structural elements to incorporate, and so it was not helpful. So I called János and discussed with him that maybe we have to purchase already modified nucleoside, which triphosphate, and then try to incorporate to the RNA. And he suggested to me that try to order from TriLink. So I selected these 10 nucleoside triphosphate, and because all of them is naturally present in our mRNA. And try to see, and routing that the T7 RNA polymerase takes that as a substrate. At least half of them was incorporated, so we generated mRNA with modification. And of course, we were curious to know that whether they will be not immunogenic. Here we tested out these in isolated human dendritic cells, which drew isolated from the blood. And what we found is that here in blue is what, the synthetic one, which I made in the tube, and then which contains uridine. And some of the others were not immunogenic, some of them were still immunogenic. And when we took a closer look, we found that which was not immunogenic, at that case, the uridine was always modified. So we thought, okay, nature recognized uridine as the immunogenic material, and if we modify it, then the RNA is not immunogenic. Of course, because we want to use it for therapeutic purposes or making vaccine, it was important that the RNA has to be translatable. And although the 2-thiouridine, we couldn't get any protein from it, but look at here, the pseudouridine, we get 10 times more protein. I mean, this is a real icing of the cake, because now I have non-immunogenic RNA, and I can get so much protein from it. So we were or at least I was very happy, because at that point, it was still considered that the conventional RNA is the good vaccine. So subsequently, actually, in 10 years later, we understand what is all about the uridine. There are actually 2 enzymes, the T2 RNase and RNase 2, which is the EDN, eosinophil-derived neurotoxin. Those are the 2 enzymes in the endosome, cuts up the RNA, conventional RNA, and release the uridine, which is the nucleoside, and that can fit right to the crystal structure, can dimerize toll-like receptor 8. So this is how our body not recognizing single-stranded RNA, and recognizing that uridine in it. Later turned out also that the TOR7 is, again, a nucleoside, the guanosine, is recognized in it. And when we did those experiments, we didn't know that our mRNA in our body has pseudouridine in the mRNA. But again, in 2014, several papers published showing that our mammalian human RNA, messenger RNA, also contains pseudouridine. It is very difficult because both of them has uracil, only the ribose and the uracil is linked differently. So the weight is the same, so that's what it was difficult to discover. So we did what we have usually in the US at that point you are doing. First you file the patent, and then you send out the paper. Then not backwards, because then you cannot have a patent. And then establish a company. We did RNARX, that you have not heard. It is all Moderna, but we were 2006, so that we have this company. And then we submitted a STTR grant, which is a small business grant, which we proposed that we will demonstrate that messenger RNA is useful when it goes for erythropoietin, is to treat anemia by increasing the hematocrit. And so we get first the 100,000, we have a milestone, and then we get a $1 million. That was the only grant in my life I got as a PI. So what happened, we did this experiment and we injected transit complex messenger RNA. Here in the red, you can see the pseudouridine containing RNA. When injected and measuring the erythropoietin level in the mice blood, we can see that it was detectable for 4 days. When we injected the protein itself, 2 hours half-life, it disappeared. When we injected the conventional RNA, it was also quickly disappeared. When we measured hematocrit a couple of days later, we could find that the mRNA translated into functional protein, hematocrit increased, and when weekly injected, we could maintain high level for 5 weeks. Of course, the important things that even in the animals, the pseudouridine-containing RNA did not induce any interferon. So this was the time again that I was forced out and I had to change and ended up in a company which had no website, but accidentally I learned about existence, and we were not in this fancy building, we were on the campus. Try to find way, because I don't speak German, and this is the Germany, the BioNTech, and with Ugur Zahin and Özlem Türeci, who established the BioNTech. So we first wrote a big paper about what you should know about mRNA and therapeutic purposes also. And then we did a lot of improvement there, you know, the RNA, the codon optimization and purification and modification. And so as a result of it, you see this small amount of protein, short period of time. Now we could get a lot of protein for extended time. So the mRNA was ready for prime time. And I will show you here 2 applications. This one, when we generated a bispecific antibody encoding mRNA. One part is seeing the immune cells, the other is the tumor cells, and we'll bring the tumor and the immune cells close proximity to eliminate. And there are different kind of bispecific antibodies in the clinic. This half-life is like 2 hours, so the patient has a pump, and every day they had to refill the pump because this short half-life. Here in animals, we did just a weekly injection of the mRNA coding for this bispecific antibody, because, as I showed you, for a couple of days, it was continuously translated. And we could eliminate already established large tumors. This bispecific antibody, as well as we have mRNA coding for cancer-specific protein recognizing antibody. So these 2 mRNA is in clinical trial. Here is the clinical trial name. Another one, which we started again, it is not vaccine, because the mRNA is not coding for antigen. Here again, we are injecting mRNA coding for 4 cytokines, and we injected in cell surface tumors in the case of animal. And what it does is makes the cold tumor hot. The immune cells will rush there, and then they recognize the tumor, and when they circulate, they can eliminate already established tumor, metastatic tumor in the lung. And this also with Sanofi is a clinical trial where patient with melanoma and head and neck cancer is injected with these 4 mRNA. While we were doing this at BioNTech, my colleague Norbert Pardy, we graduated from the same university. Actually, we are from the same small town, and his grandfather and my father work in the same butcher shop, so I know him very well. And so I left my laboratory at Penn for him, and then he started to use messenger RNA for vaccination, but now he used the nucleoside-modified one. Originally, the idea was that some of the mRNA will code for different kind of cytokines too, because in the vaccine you need an immune stimulant, some kind of adjuvant. But then turned out that you don't need that, because you know, this lipid nanoparticle, one component is actually the adjuvant itself. But at that point, we didn't know that. So this mRNA was a new virus in 2017, was important, the Zika Zika virus. And so this was this pre-membrane envelope protein coding mRNA was made, nucleoside modified, and formulated the same lipid nanoparticle as the BioNTech Pfizer vaccine. This was made by Acuitas. So it was very similar, just only the coding sequence and some untranslated region were different. And what was important, of course, we know that the lipid nanoparticle center is, of course, Vancouver, because all of the companies are there. From the liposome company, later it was Tecmira, and Acuitas is there. So they come up with this formulation. So this is very critical and as important as the mRNA in the vaccine. So Norbert generated this mRNA with a lipid nanoparticle injected to the monkeys, and very small amount already protected the monkeys from Zika viral infection. This paper published in Nature, and it was by Bial, was selected as the most important paper in the last 10 years. So this is how we get to the vaccine development here, and you already know that 2020 we learned sequence information about the coronavirus, and on the same year, we get the vaccines. Just to mention two things, that people who thought that that was the first time nucleoside modified mRNA was injected into human, I can say that in 2018 already was injected to the heart. AstraZeneca, Moderna worked on it, and a patient who had a bypass surgery, they injected to the heart muscle, and this VEGF-A mRNA codes for a protein which generate new blood vessels, and the phase 2 trial just finished this year with a good outcome. Another application was when it seems like using Cas9 mRNA. This was 1-methylpseudouridine containing RNA used intalia to treat a patient who has amyloidosis. This patient, when they are diagnosed, they usually 5 years later they die. But the reason is because they make a toxic protein, and what this technology did, delivering the guide RNA together with the Cas9 mRNA, they could successfully interrupt in the hepatocytes this genome where this toxic protein is made. And this patient, even one therapy, one injection, and 1.5 years later, still they are not doing this toxic protein. There are many, many things you can daily hear about different vaccines, different application of the mRNA, coding for secreted protein, internal protein, and others. And the future is here somewhat, and for those who are working on the formulation. So with that, I would like to say thanks to colleagues. I had, I didn't have a postdocs or a graduate student when I was at Penn, but here at BioNTech, and you see that they wear at least gloves. And I am also very grateful to my family. To the girls here, I can say that, and I like to brag, my daughter is a 2-time Olympic champion rower. And of course, without my husband, because behind every successful man, there is a hardworking woman, but behind me also there my husband who supported me and let me work even the weekends. And with that, thank you very much for your attention.
So we will have time for a few questions, and then if people want to stay for an informal discussion, that will be fine. But if you have questions right now, please use the microphones on either side so that we can capture the questions.
It seems everything was clear.
Very.
No question.
I'm sure somebody has a question.
It was a lovely talk. Can you I'm sure it's understood, but can you just explain to the people in the audience why is it that endogenous uridine doesn't cause an immune response if it's only the uridine that comes in the context of an exogenous source?
When you have an injury, you have response. But as I told you, even when we isolate total RNA out of the mammalian cells, because it has modification, you don't get as much response when those mRNA, which would have zero modification. But you are right. When we injure, then we have a sterile inflammation, so that our RNA also activate. I showed when we isolate. So, of course.
Is it thought that the scale of the RNA that's added to cells in the context of a transfection or something, that's part of it, I guess?
So what happened is that when we deliver a particle which contains the material, it is picked up mostly by macrophages and dendritic cells.
Right.
Because they have the endosomal activity.
Right.
And T cells, you cannot transfect at all.
Right.
And even the muscle cell is not getting, because they are not doing this. There is not their job to pick up things, so that all these immune cells has actually also these two enzymes, and these two enzymes is very critical because they just work when a low pH is present. And if you want to generate a permanent cell line, you just have to knock out actually RNAs, T2 RNAs. So it is important, because if that is not present, even you don't generate from your own injury immune response.
Thank you.
Vivian?
Thank you, Ron. Beautiful talk. With so many therapeutics in the pipeline, how will you target them with those LNPs to the specific tissues?
Yes, so we already published earlier that just changing even the ratio, same composition, but the ratio of the different lipid, positively, negatively charged lipid, you can redirect the particle to the spleen or to the lung. In the lung, of course, because the endothelial cell will pick it up, and it's most endothelial cell, most blood vessel in the lung. But targeting also happening with antibodies. Of course, the particle you cannot freeze together with the antibodies, so it had to be reassembled before injection. One of them, Dan Peer in Tel Aviv, he's doing that, incorporates some kind of protein like the protein G, which would bind to FC region of the antibodies, and then before injecting, they put together the antibody and the particle with the RNA, and injecting and delivering. And so there are also, if you don't want to certain cell type, certain organ to your RNA to translate, for example, Moderna is using particles injecting to the tumor. And then the particle can circulate maybe and get to the liver. They don't want to translate in the liver. So in the mRNA, they incorporate the 3' UTR miR-122 target site, which is the hepatocyte overexpressing miR site. And then when it enters to the hepatocyte, that RNA, it chop up, and then you don't get protein. So there are, you can refine the translational how you want to translate your RNA. But also there are publications where certain ligands was incorporated to the particle, and then certain cell types will pick it up. In the case of T cells, I mentioned that there are no endosome, they will not pick it up. So in that case, they put a ligand to it, which the T cell has a receptor, and is known that it will bring inside the cell, and that's what is used. So a lot of tricks already published, and more is needed, of course.
Okay, so there first, and then Jennifer.
I wanted to ask specifically if you see any future in using mRNA vaccines for treating various kinds of neurological and mental health conditions. So one of the problems that we have with a lot of these conditions is that they involve neurotransmitter imbalances, but the problem is that the imbalance is in a specific region. And so, for instance, like with treating depression, we can give serotonin reuptake inhibitors, But part of the reason they have so many awful side effects is because basically you're increasing the amount of serotonin everywhere in the brain and not just the specific target sites where you're getting the imbalance. And I'm wondering, and it's currently a really difficult problem to try to figure out how can we target like specific regions, for instance, of like the dopaminergic system or the serotonin system. Do you see any possibility there?
So this wouldn't be vaccine. You know, you have to call it vaccine when the antigen. So it is therapeutic. You mentioned serotonin, and I don't know that this is a protein and how long or peptide. You know that because there is a problem. I try to make, for example, endorphin encoding. mRNA, and if it is something shorter than 100 amino acid, you don't get secretion, because it was had to be pre-molecule or longer peptide, longer protein and chopped up with peptide, because it won't get out from the system. But let's say that's a regular protein, and you want the secretion to increase. I mean, one problem with RNA is that it won't go through different layers of cells. It won't go through one cell and another. It doesn't have that kind of ability that like maybe some virus would have. So locally, right now, is, you know, with the needle, you will inject where you want or deliver there. But, you know, if more advancement is coming with targeting those tissues, then you could try it. We did the intrathecal, of course, neurosurgery. I had to do something to relate to that, so we did intrathecal. We injected to the brain, and what happened is every time is the mRNA was taken up by the epithelia, which lined up the tissue. It won't go through layers of, and it won't end up certain neurons which you want or wish. But maybe, you know, here you are guys here, and then come up with some formulation.
Thank you.
Jennifer?
Yeah, thank you for the wonderful talk. My question is, would there be additional challenges associated with the delivery and effectiveness of non-coding RNA therapeutics like tRNA versus mRNA?
I mean, we are not working in tRNA therapy, so we're working with all of this is coding mRNA therapy. So what do you want to do with the tRNA?
So there have been a couple of initial studies using suppressor tRNAs to target premature stop codons that can cause diseases. So I was wondering if, because you don't have things like the cap for stability, if there would be additional challenges.
Yeah, so actually we also worked on, and this year we published on how we could make some stop codon read through and other things, because there was a publication that if you incorporate pseudouridine in the stop codon, then it will read through. We could not find that. We found that stop codon actually has higher fidelity recognition if it is nucleoside modified. The pseudouridine was there. But yeah, so that I don't work with tRNAs, but you know, I will, on my flight back, I will think what it should be changed on that.
Thank you.
Okay. Go ahead.
Thank you so much. I just have a few closing remarks on behalf of the university. Thank you. You don't need to go far. On behalf of the university, I would like to thank our distinguished Canada Gairdner International Award laureates for coming to York University today to tell us about their journeys in research. It really has been wonderful for us to hear your stories. I'd also like to thank our audience and guests who joined us today to hear about the research, and it's wonderful that we are able to be here again in person in 3 dimensions. I'd also like to once again thank our sponsors, without whom an event like this could not take place. Thank you to the Globe and Mail, the Government of Canada, CIHR, York University, and the Gairdner Foundation. And finally, I would like to thank everyone who worked so hard behind the scenes to make today happen so smoothly. And I do have a very small memento from York University that I would like to present to Drs. Karikó and Dick on behalf of the York community, if you don't mind coming up. And I do hope this doesn't take too much room in your suitcase on your way home. Thank you so much. Thank you.
Thank you very much.
And as Dr. Perlin had mentioned earlier, at this time, our distinguished speakers have generously offered to spend some time speaking with trainees, but we will also allow faculty members and others, yes, Vivian, you can stay behind, and have a smaller, more informal discussion for just a few minutes. They have just about 10 or 15 minutes before they have to leave. So I will close the session and say once again, thank you to everyone for today. Thank you.